Good afternoon, everyone, and thank you for joining the webinar this afternoon. My name is Meredith James. I'm the COO of Flagship. Today, we're going to be talking about biomarker strategies and implementation in drug development with Susanna Corey-Torrey. Susanna has been in the regulatory space for over 30 years with multiple degrees and experience across the industry, including the agency as well. So welcome, Susanna. Thank you for taking the time to speak with us today.
Thank you, Meredith, and hello, everyone. It's a great pleasure to be with you today to discuss one quite complex and ever-evolving topic of biomarkers in the context of drug development. Flagship recently expanded its services to include regulatory and drug development support, and this webinar is to introduce you to the services and what we can do for our clients. We can take your program at any point of development and develop it further up to the endpoint, whichever that endpoint may be. We work on a principle of partnership with our clients, and we understand the regulatory standards and requirements and pathways, and we make sure that we apply them for a time and cost-efficient development. We have a successful track record with the agency, and we can interface and present you with the agency. So all of these aspects are included in our services. Today, we will provide a broad overview of the biomarkers and their implementation in drug development strategy with a focus on drug development, because there are other uses of the biomarkers. And by no means this is not exhaustive presentation, but we welcome you to contact us after this webinar if you want to learn more about the details and how we can help you with their programs. We will discuss the biomarker definitions and categories, how strategically to implement biomarker and their SS evaluation into drug development and clinical trials, and what the key regulatory points and to consider for approval pathway may be. Always eternal topic, nice to have versus need to have, of course. Biomarker regulatory definition is a characteristic that is measured as an indicator of normal biological processes or pathogenic processes or responses to an exposure or intervention, including therapeutic interventions. They can be of different nature, molecular, histological, radiographic, physiological characteristics, and they can present single characteristic or can be a panel of multiple characteristics and can be biomarker signature. And they can be qualified via regulatory process for use across development programs. Biomarker assay or test is actually just not a biomarker, but also entire set of reagents, assays, and methods for detection or measuring the biomarker, and they are validated analytically and evaluated and qualified clinically. They are very important tools for successful biopharmaceutical targeted drug development and approval, with the context of use as a critical driver for such development. Biomarkers can be involved in just any phase of targeted drug development, and they can contribute to the success of drug development at any phase. They can serve as a molecular target for drug action or support the drug action. They can be part of important pharmacodynamic response. They can help, they can help the evaluation of safety, and they can serve for monitoring of important outcomes in the trial and in contributing to the overall characterization of risk-benefit profile of the drug or therapeutic intervention. Among the other factors, the probability of drug development success has been shown consistently to depend on development and use of biomarkers and associated technologies, resulting in increased efficacy, improved safety, and more personalized treatment. As much as we would wish, there is no such a thing as ideal biomarker from drug development perspective. However, certain attributes of a biomarker are highly desired. For example, the biomarker should be part of the disease or the pathobiology, either related to disease burden or disease spread and other characteristics like metastatic spread of tumors. Ideally, such biomarkers should increase pathologically in the presence of disease related to high sensitivity, or does not increase in the absence of disease associated with high specificity. It should change in accordance with the clinical evolution, reflecting the status of disease or the treatment, and it should be measured by accompanying laboratory assay that is robust, easily reproducible and reliable, and certainly economically viable. Should be relatively inexpensive, and it should not add to financial burden to the patient. The goals of biomarker use changes with drug development phases, and in early phase development, the focus is really on the biomarker and how biomarker supports the outcomes regarding target validation. It can help early compound screening, part of pharmacodynamic studies and response, and actually can serve as exploratory endpoints. So the focus is on really drug mechanism readout, as shown on the graphic on the right side of the slide. In the late phase development, the focus shifts toward diagnostic and outcomes measures, and biomarkers are increasingly used in this later phase development to support study eligibility, patient eligibility, stratification factor, or even serve as a surrogate endpoints. There are different biomarkers categories, and they're all related to the context of their intended use. This slide is very busy, and I will not go into details. You will be provided the presentation after the webinar, and you can inform yourself with more details. I will just provide a brief summary of the major biomarker categories, like diagnostics, prognostic, and they can be predictive. They can be part of pharmacodynamic response. They can be used for monitoring, to assess susceptibility to risk, and of course, they can be used for measuring some important safety parameters. All of these biomarkers can be used in research and drug development. However, in order to them to be support of a regulatory process and become eventually part of drug labeling, their use must be indicative of their potential to be reasonably useful or directly useful as a surrogate endpoints in a reliable and reproducible manner, providing sufficient clinical evidence on predicting specific clinical benefit. So how we position implementation of biomarkers in clinical trials and in drug development, this slide might be really the most important summary we provide in the context of this presentation. There are several points to consider. First of all, the biomarker role in the development of a particular therapeutic or in clinical trial needs to be defined. Key questions we ask in this phase is, does the selected biomarker adequately supports the clinical drug evaluation? And do we have a biomarker test ready for use within a trial to adequately support the outcomes of that trial? In this phase, maybe we can rely on information just coming from preclinical testing or from discovery phase, and that information can be quite limited. So we need to plan for biomarker implementation in a stepwise and data-driven approach. That include developing scientific rationales, understanding desired assay characteristics and how we incorporate in the trial designs. And it's a similar process how we assess therapeutic readiness for clinical evaluation. How is the drug ready for clinical evaluation? So the selection of a fit for purpose biomarker assay and lab vendor are critical in this process and requires really careful coordination of all available expertise, capabilities, and resources. If you are, for example, mid-size and large pharma company, that means that you will carefully assess balance between your in-house capabilities and resources and what you need to outsource. However, if you're a small or emerging biopharma, then you will most likely outsource most of these activities, and selection of right vendor is a critical factor for success. We really invite all of you which are in this situation to contact us and discuss details of your needs. Analytical, we support both smaller and larger companies, and our capabilities can be quite versatile, so please contact us. Analytical validation in the next phase of the biomarker assay is always specific within a context of clinical use, and you should make sure that this is adequate for the purpose and the clinical trial setting. This also can include additional considerations, selection of best tissue matrix, what is the nature of the biological sample, what is the ease of obtaining the biosample from patients, and how you properly handle the samples, what is the physical chemical characteristic of the biomarker, and what kind of reagents you need to plan in development of the biomarker assay. These are really important questions that can have a very practical impact on what you're doing. This process can progress to different levels depending on the phase of overall development, and you may not have to have a full development of the assay early on, but you definitely need to have a strategic understanding how that may develop as the drug development progresses. So in the earlier phases, we usually have limited validation of the assays, and as the drug development progresses and it's showing being more successful, your assay also needs to follow. This is important, especially to support pivotal clinical trials and regulatory approval. The whole process therefore requires early strategic planning and understanding, and continuous data review and planning for further investigations, including what regulatory pathways are available as an option, and consideration for companion diagnostic. That's a challenging one because that might be not amenable for the earlier phases of development, but suddenly we have accelerated development and you have a need to develop your assay as a companion diagnostic. From regulatory perspective, there are two main regulatory pathways to integrate biomarkers in drug development and subsequent clinical use. They can be developed as an integral part of drug approval process in specific clinical indication. And the emphasis here is in specific clinical indication, or it might be different indications depending on the type of the regulatory pathway, and we'll touch that briefly a little bit later on. this incorporation of the biomarker in drug development pathway is more common, both for new biomarkers and for new uses of established biomarkers. Or biomarker can be formally evaluated via FDA biomarker qualification program to support multiple drug development programs. These two processes are never isolated from each other, and many activities in parallel are undertaken. Development of biomarkers supporting specific drug development program is usually responsibility of the sponsor. Their incorporation in the drug development program is usually responsibility of the sponsor. However, if new information suggests their utility in other drug development programs, then FDA may include their use in their regulatory guidances or approved product labeling. Each biomarker is qualified for a specific context of use, and it's important to note that this is the biomarker that's been qualified in this process, not the assay. The biomarker assays can be used to detect, quantify, and or monitor individual biomarkers or a group of biomarkers, like a biomarker panel, where the most appropriate platform depends on the matrix, analyte, and specific biological question to be answered. If biomarker has integral role, such as one that is used for patient eligibility for a clinical trials, such as may need to be performed under FDA oversight, depending on the inherence or associated risk, and if their results will be reported to the patient or his or her physician, the assay has to follow, it has to be performed in a CLIA certified laboratory. Fit for purpose of the biomarker assay, the rigor of its analytical validation and need for regulatory oversight are critically linked to its clinical utility. An assay developed for one fit for purpose may not be adequate for another. The analytical validation of the biomarker assay is complex. It's a multi-layer process with main goal to establish that the assay performance characteristics are acceptable for intended context of use. This is also a busy slide. I'll just go through the main points. This process includes development of an appropriate assay, utilizing proper biological matrices, characterizing the assay sensitivity, specificity, reliability, and reproducibility to support definition of assay acceptance criteria. Assays from that perspective can be developed as qualitative, semi-quantitative, or quantitative. The level of their validation can be exploratory, can be more advanced, but still as in-house assay, and can go all the way through full validation. Important factors can be variability of measurements and sources of their variability, the intended use, the sample collection and processing and logistics around it, as well as patient selection. All of these factors are important that can impact in the individual analytical assay validation. The analytical assay validation is intimately interrelated with its clinical evaluation and validation. And clinical evaluation validation of an assay has a goal to establish that the correlation between the biomarker and the clinical outcome of interest is acceptable for the proposed context of use. Different drug development phases call for different levels of biomarker assay development or validation. Therefore, we ask different questions at each of these phases. In the discovery or preclinical phase, it's all about the establishing scientific biomarker validation, its role and its potential utility for further investigation. But the appropriate assay doesn't need to be really validated. It's not required to be validated, nor there is a sufficient information, most likely, to support this validation. Typically, you may use screening assay in this phase. In the early development phases, up to the pre-proof of concept and proof of concept, the focus is on establishing actually proof of concept. Qualification of the assay for the validated biomarker may be required for desired use, but still full validation may not be needed. And it depends on strategically how you position the development of biomarker and biomarker assay. You may have had all resources. You are pretty sure that your drug will be successful in clinical evaluation, so you invest early into it. But if you don't, then one of the risk mitigation is really to carefully tailor how much you validate, characterize the biomarker assay in these early phases. Once you have, and this is all logical, the risk or failure for drugs in this phase is still high. And it's getting less as the clinical development progresses. But especially in phase 1 and phase 2, you really don't want to invest unless it's necessary in biomarker assay validation. Once the proof of context is established in later development phase, the qualification of the biomarker assay is required, and its validation is highly desired and needed, actually, and your investment in the biomarker assay at this point of time will increase. If the assay is essential for safe and effective use of the drug after the drug is approved, then the assay will proceed in development of companion diagnostic with all resources and capabilities that are required to do so. So biomarkers have become really integral part of precision-based targeted drug development and clinical studies, and there are different ways how to implement them in the clinical study designs. This is nowadays done through use of so-called SMART or master trial protocols as early in the clinical drug development as possible, especially in oncology. Major study designs of this sort may include umbrella type of study protocols to study multiple targeted therapies in the context of a single disease, usually characterized by a particular biomarker or biomarker signature. Also, there's so-called platform trial designs that use adaptive designs and can study multiple therapies in the context of a single disease with some biomarker characteristics, but allowing these therapies to enter or leave the trial as new data become available, and increasingly used basket clinical trials to test practically therapy among patients that share a common biomarker across different disease types, such as tumors. This slide shows you the graphical illustration of this main trial designs. Biomarkers are deeply inherently embedded in these trial designs, and as this slide shows, all key points to consider when designing, let's say, umbrella or basket trials include biomarker considerations, and these considerations can include biomarker prevalence, biosample collection and associated logistics, accuracy of biomarker assay, and ability to establish cutoff points to support sample size calculation and randomization. So it's really inherent integration into clinical trial design of the biomarkers. This type of clinical trial design utilization of the biomarkers caused, and it's increasingly causing a biomarker-driven shift from traditional drug approval based on a disease toward biomarker-based or tissue-agnostic regulatory approval. accelerating greatly drug development process. For example, you can see on the infographic, traditional drug approval process in oncology is based on tumor type, and clinical development follow traditional sequence of clinical phases, resulting in regulatory approval. That process can be long and quite expensive, and can include substantial investments on the sponsor side, as well as greater risk because the drugs still can fail in phase 3 pivotal clinical trials. The biomarker-driven regulatory pathways include biomarker-defined subpopulations for evaluation of the drug effectiveness. And now we have opportunity that the drug gets conditional approval much earlier than phase 3. And that can be as early as expanded phase 1 or 2, depending on the magnitude of therapeutic effect. And in third scenario, it's possible now to get accelerated drug approval targeting practically biomarker across different diseases or tumor types. The regulatory approval of pembrolizumab for treatment of adult and pediatric patients with unresectable or metastatic solid tumors with microsatellite instability, high or mismatch repair deficient genetic markers was the first accelerated disease or tissue agnostic regulatory approval. It really signaled new era in oncology drug development. There are currently several categories of biomarker assays and definitions to support effective drug development. This is also very busy slide. I'll just go through the main categories. We have in vitro diagnostic kit that contains all reagents, instruments, methods, which intended uses to diagnose disease or condition, including health status determination in order to cure, mitigate, treat, or prevent disease or its sequelae. IVDs are used for collection, preparation, and examination of specimens taken from human body. Laboratory test is a type or subtype of IVD that is designed, manufactured, and used within a single laboratory. Clinical trial assay is also a subtype of IVD that may be used in drug development to support patient selection or prediction of therapeutic benefit. This type of test may involve further into development of companion diagnostic, which is IVD that provides information that is essential for the safe and effective use of a given therapeutic product. This use is stipulated in instructions for use in the labeling of both the IVD and the therapeutic product. And complementary diagnostic device adds in the benefit-risk decision-making about the use of a given drug when that difference is clinically meaningful. They help to identify, for example, patients that respond to the drug better. Information of such device can be also included in the drug labeling. Laboratory developed tests are subsets also of IVDs intended for clinical use and are designed, as I said, manufactured, used within single laboratory. Depending on the drug development being global or limited to one geographical or regulatory region, one needs to understand whether investing into laboratory developed test is the best investment for your overall program, or just for a particular clinical phase. Laboratory developed tests are commonly validated under CLIA regulations. Historically, they were not subject of FDA oversight. However, that climate changes, and it's still still subject of substantial debate, resulting in FDA not really finalizing their guidance on laboratory developed tests. Depending on the character of the disease and associated risk with the test, FDA may request and will request a review of the information about the test, its design, validation, and performance characteristics. This is mostly safety driven, and it is, for example, can be for a serious disease to treat life-threatening condition, drug for a serious disease that can be life-threatening, or there's some inherent risk for the results of the laboratory developed test translated into patient well-being. The regulatory labeling and distribution of IVDs or CDX can differ according to their regulatory approval status, the approval status of the drug, of their own approval status. They can be labeled as a research use only or investigational use only. The research use only label implies that IVD is not for clinical diagnostic use. It is in the phase of evaluation of assay design and its performance, and as such, it contributes to development of knowledge about disease. The investigational use only refers to the fact that IVD is under performance evaluation, and it is used for diagnosis or treatment decisions, or can be used for eligibility in a clinical trial. This type of label also typically meet the criteria for investigational device exemption because inherently it's considered to be of higher risk. Companion diagnostic can be lab-based or commercially distributed. The lab-based companion diagnostic are developed completely by a single laboratory, and a CLIA compliant laboratory is required to file either pre-market approval or 510K application, but the approval is also linked exclusively to that single lab to deliver the results. In contrast, commercially distributed companion diagnostics are first developed by a single manufacturer, approved by FDA, and then these devices are made commercially available and supplied to clinical testing laboratories. Clinical testing can be performed in any appropriate laboratory that analytically validates use of the assay under their lab conditions. The regulatory oversight of IVDs is a risk-based system. With this regard, there are two main risk categories, significant or non-significant. Significant risk device or IVD, because IVDs from regulatory point of view are considered as devices, presents a significant risk to health, safety, and a welfare of a subject. And it's important to note that it is potential of occurrence of significant risk that matters here, not the likelihood of the occurrence. The risks may include risk associated with collection of tissue samples via procedures, invasive procedures like tissue biopsies, but also risk that is associated with making erroneous decision for patient selection or who will receive drug or not, due to the rate of false positive and erroneous rate of false positive, false negative results. If the drug is toxic and the patient is, let's say, false positive for the biomarker, then that means that that patient will be necessarily exposed to substantial toxicity from the drug, and that's really not acceptable. So therefore, this type of IVDs will require submission to assess if the IVD might be exempt or not from investigation. And that's also a process that will require planning and also performing trials. It will result in approval clearance by FDA of the IVD. The non-significant risk IVDs are those that do not require FDA approval prior to clinical use. And FDA doesn't need to be noted in this situation as long as the risk associated with the use of the IVD during drug development does not change. If changes, then the sponsor is advised to notify the FDA and discuss the next steps, because FDA can impose clinical hold on the use of the IVD in clinical trial and practically recommend to the sponsor to initiate the investigational device exempt process and submission. That may result in practically changing of the overall program timelines and including additional investments and resources. There are three main regulatory pathways how one new IVD or companion diagnostic will be approved. One of the pathways is so-called 510K, and it is appropriate when the new diagnostic device can be shown to be equivalent to an existing test that is already on the market. We call it a predicate test or device. And the focus on 510K submission is really on establishing and demonstrating the equivalence with predicate, and the content of the studies required is not really comprehensive as that one required for the PMA, and in many cases the studies may not be required at all. For the pre-market approval pathway is for new diagnostic devices that will be used for critical decision on diagnosis, treatment, or medical management, and for which there is no predicate test on the market. The content of the PMA submission is very structured, and FDA regulatory guidelines prescribed the content of type of required studies that can be substantial, and it resembles in a way to the application of new drug. And there is a third pathway called de novo reclassification. If no predicate IVD exists, but the new IVD is of low or moderate risk. At the end, there's so much to be said about the biomarker and their implementation in drug development. It's really not easy to navigate the development, the regulatory maze. This might be the drug development process and process of development of biomarker assay IVD toward companion diagnostic may schematically be presented as a linear sequential process, but in practice, it really resembles like a maze. And we at Flagship can assist you with any of the major activities related to biomarkers, biomarker assays development. At any phase of drug development, we can help you to demonstrate feasibility, to develop a prototype assay. We'll support analytical validation and clinical validation of the assay, and we'll support your registration process all the way through the launch, and we can also help with the commercial support and commercial aspects. Now we have also internal capabilities to prepare the regulatory submissions and to file to agency, and we can support you also in your dialogue with the agency. Across all of these phases, one important thing is really to engage in dialogue with the agency as early as possible, having a clear understanding what the strategy of development is, and then apply that strategy in a stepwise data-driven process. That will be all for today, and I truly appreciate. Thank you for listening. We are now ready to take questions. And just to say one more time, it's been great pleasure to discuss with you this topic. And please contact us if you have any question regarding the presentation or regarding your programs. We're happy to schedule consultation and to understand how we can help you with your development efforts. Thank you very much again.
Well, thank you, Susanna. That was great. So let me start running through some of these questions. The first question that we have is, can you elaborate on the risk mitigation strategy with companion diagnostic development?
This is definitely very complex topics, but as I just emphasized, having an early strategy of biomarker assay development in the context of drug development, it's really critical. That means that one needs to understand at which phase of drug development, what kind of a level of validation of the assay may be needed. And there are many factors that can influence at points where we can mitigate the risk. It's very important to, for example, have understanding at which point of drug development the IVD, let's say clinical trial assay, may lead into companion diagnostic, and what will be required to provide such a relatively short and simple transition. One of the things, for example, is how we incorporate the biomarker evaluation into the study design. Do we have prospectively defined eventually endpoints? Did we collect prospectively the tissue samples? Did we retain the tissue samples or other biological samples? That retention of samples can be critical, especially for the transition from CTA to companion diagnostic. So there are a lot of things that will need to be clearly delineated in the strategy, and the strategy needs to include points of inflection, including go/no-go decisions, but carefully tailored as the drug successfully progresses in the development process, and be ready to make transition.
Great, thank you, Susanna. And obviously, as Susanna had mentioned earlier, we're happy to help you to review strategy and risk mitigation. Just reach out and we'll be happy to have that consultation. All right, Susanna, what are some lessons learned from past biopsy assay development efforts?
There definitely each assay has some lesson to be learned. But when we're talking about biopsies as a way of collecting the tissue samples to support biomarker assay development and use in the context of clinical trial and clinical use, is to understand first if this is really the best assay for your biomarker. Depending on the type of tumor, for example, do you have opportunity to do repeated biopsies? I will give you just an example. If you develop a drug in a brain tumor indication, one needs to understand that possibilities for repeated biopsies are practically very low. Also, if you have a tumor that really doesn't provide good quality of tissue samples because it's a very aggressive metastatic, like small cell lung cancer, you may have biopsy as a collection method, but the quality of the tissue you may get may not be really the best to support biomarker assay and evaluation of the results obtained with such assay. Also, if you have, let's say, global trial, and then you need to understand the logistics and the capabilities of the labs to collect, process, and prepare the samples for shipment, let's say, to the central lab. Nowadays, especially with pandemics, these things are really not that easy. Also, the assay needs to be adequate to the physicochemical characteristics of the biomarker in these tissue samples obtained by biopsies. So you need to understand that all reagents are appropriate, and your reagents do not in any way negatively impact the ability to detect the biomarker in the tissue obtained via biopsy. You may also need to consider an alternative strategy to biopsy. One of the emerging and expanding alternative methodologies are circulating tumor cells. However, they also are associated with questions, and as of now, they might be good to be used for monitoring of the treatment response or the disease evolution, but they might not be really for diagnosis. But it's absolutely a technology that requires just a blood sample collection, and it's certainly not that invasive compared to the tissue biopsies. There are many, many other factors, but these are the practical questions that one needs to understand before initiation of a clinical trial, for example. Meredith, do you want to add from your perspective?
Yeah, no, absolutely. Thanks, Susan. Obviously, one of the additional challenges you can run into that you alluded to is logistics. So making sure that you have appropriate instructions, ahead of time in the lab manual. What does that look like from a site perspective so that, again, you're getting the best quality tissue to ensure the results going forward. And again, Flagship has a lot of experience with helping to generate this information and really to help support your trial so that we can aim to get the best data and the best answer for you. So, all right. So actually, Suzanne, I may answer this one. This one should be pretty straightforward. Just came in. What orthogonal method do you routinely use to assess IHC? And obviously, they threw a couple of things out there. Obviously, at the end of the day, it will really depend on what it is that you're trying to measure. I will say that routinely here we use things like nanostring, PCR, Western on occasion, although obviously there's some challenges that come with that. But one of the unique things about Flagship is that we have a very integrated scientific team, and so they'll sit down with you, review your assay and your biomarker, and really try to figure out what makes sense in the context of that to ensure that we're actually selecting the most appropriate orthogonal method. And of course, we have a lot of options there. So I don't know, Suzanne, if you wanted to add anything.
No, I think you really said it all. And it depends. It is really, on case by case. So we really welcome to contact us for additional consultation to understand which method might be the best. And also to look what is out there available already, previous experiences and so on.
All right. So Susanna, next question. It would be great to hear more about follow-on companion diagnostics in LDTs.
Well, the follow-on diagnostics, IVDs or companion diagnostics, practically developed for an approved indication that's been originally approved with the original companion diagnostic. And the challenges and actually goal how to evaluate this diagnostic is how to show that this new IVD is equivalent to the originally approved IVD or companion diagnostic, so it can be used safely and effectively in the clinic. And that process may not be straightforward, has challenges, and the possibility to do a clinical trial may not be an option or may not be ethical. Also, the sample retention that I emphasized during my presentation can be really very helpful in this situation, and a planning of external accordance study can be supported by that. In some cases, thorough analytical comparison of analytical performance of the new IVD versus the originally approved can be sufficient. It depends what the complexity of the IVD and its use is.
All right, thank you, Susanna. I think that's all the questions that we have so far. If anyone else has a question, go ahead and submit it. We'll give everyone just one more minute. You can always ask questions after the fact too. Feel free to reach out to us, and myself and Susanna will be available at any time to follow up with you. Again, Susanna, I can't thank you enough for the information today. I think it was really helpful, and I hope everyone has a great afternoon.
Thank you.